See today chromosomal aberration. So, have you heard aberration or chromosomal aberration? What are chromosomal aberration? Have you heard? Okay, firstly, we will talk about chromosome. As you know, these are tiny thread-like structure found in the nucleus of most living cell. What they carry the genetic information in the form of gene. So these are made up of protein. They are actually what are the protein that are basically forming the chromosome? They are the histone protein and the DNA. And each person normally have 23 pair of chromosome. They are 26 in number. As you can see, this is a chromosome structure. These are the two chromatid and this is the centromere. Now, chromosomal aberration. See, mutation that cause change in the structure or number of the chromosome. Any type of mutation that is changing the structure or number of chromosome, they are called chromosomal aberration. And this generally the incidence of chromosomal abnormalities in is 5 to 6% per thousand. If you will see the chromosomal abnormalities in 1000%, so out of 1000%, 5 to 6% they are affected by chromosomal abnormalities. Many children with chromosomal abnormality, they have mental or physical defect. Now, earlier we also talked about this, that is aneuploidy or non-disjunction that we have seen in the first or second class. So this particular, if your non-disjunction takes place in first meiotic division or if non-disjunction takes place in second meiotic division, so what will happen? See, if there is a first division non-disjunction, firstly these chromosome they have to separate it into two different cell, but they are not separating into two different cell, they are coming in the same cell. So it is because of first meiotic non-disjunction. So as you can see, they are forming the trisomic and monosomic condition. So that is creating non-disjunction. That is, sorry, that is creating aneuploidy. Due to this non-disjunction, due to this meiotic non-disjunction, we are getting the aneuploidy in the zygote. Now see if their non-disjunction takes place in second meiotic division. So this is the first meiotic division, this is the second meiotic division. Now see, in the first meiotic division, the chromosome, they are separated easily into two different cell. Now the chromatid, they have to separate into two different cell, but here two chromatid they separated easily into two different cell, but here same two chromatid they are coming in the same cell. So they are forming the trisomic, monosomic, as well as disomic condition. Disomic, that is normal, trisomic abnormal, monosomic, that is also abnormal. So we are getting normal as well as abnormal gamete or progeny in the, if there is a non-disjunction in second meiotic division, we are getting normal abnormal in second meiotic non-disjunction. We are getting all the abnormal gamete or all the abnormal progeny in the first meiotic non-disjunction. Is it clear?
Yes, ma'am.
Okay. Now types of chromosomal abnormality. See, if there is abnormality in the structure of the chromosome, that will create the structural abnormalities that involve deletion, duplication, inversion, translocation. If there is abnormalities that is related to the numerical, uh, that will change the number of the chromosome, that is numerical abnormality, that is aneuploidy, polyploidy. Now we are going to see the numerical aberration first. So as you know, this chromosome, they are actually 46 in number. So whenever there is loss or gain of chromosome, that will create the numerical aberration. It include both autosomes and sex chromosome. Now types of numerical aberration. See, change in the number of chromosome, that is also termed as ploidy. As you know that if there is number of chromosome, if there is a change in number of chromosome, how many number of chromosome are there? 46. So if the number is where the number will vary, that is termed as ploidy. So there are two kinds of ploidy, euploidy and aneuploidy. Now firstly, euploidy, that is the state of having exact number of haploid chromosome, that is 23 pair of or multiple of haploid chromosome. So it involved variation in the number of complete sets of the chromosome. If you will see the variation in the complete set of chromosome, that is euploidy. That is, see, monoploidy, how there is variation in the number of the chromosome, or how in the variation of complete set of chromosome we are going to see. So there types of euploidy, there is monoploidy. So condition in which each chromosome is represented only once, that is having 23 number of chromosome, that is monoploidy. So in some cases it occurs naturally, but in some it occurs due to parthenogenesis, that is reproduction from an ovum without fertilization, especially as a normal process in some invertebrate and lower plant. Now diploidy, as you know, if you will represent the progeny by n, that is monoploidy. Sorry, unable to write. If you will represent the progeny with n, that is monoploidy. If you will represent the progeny with 2 n, that is diploidy. So condition in which organism have two set of chromosome or multiple of haploid chromosome, that is diploidy. So all sexually reproducing organism have two sets of chromosomes. So most species of animal, they are diploid. Now polyploid, condition of having more than two set of chromosome, like 3n, 4n, 5n, 6n. 3n means triploid, 4n tetraploid, 5n pentaploid, 6n hexaploid, and so on. So some animals such as lizard, amphibian, and fish, they are polyploid in nature. Now aneuploidy, that is loss or addition of one or more chromosome to the complete set of the chromosome, that may be extra 47 or 45, that is aneuploidy. See, it was first discovered by Albert and his colleagues in 1920, that caused by failure of chromosome to separate during meiosis. As you know, that chromosome, they separate during cell division, and if there is failure in that particular separation of meiosis, so that will create aneuploidy. Now hyperploidy, a state in which cells contain one or more fewer chromosome than what is normal, that is hyperploidy. Hypo means lower, that is less in number, one or more fewer chromosome, then that will create hyperploidy. And it include monosomy, nullisomy. Monosomy, that if loss of one chromosome from whole set of chromosome. See, loss of one chromosome, remember that from whole set of chromosome, that is monosomy. But if there is a loss of chromosome pair, not chromosome, loss of chromosome pair from whole set, that is known as nullisomy. Nullisomy means 2n minus 2 condition, means two chromosome, they are lost but they are in pair. One chromosome is lost but it is not in pair, it is loss of one chromosome from this whole set of chromosome, that is monosomy. And 2n minus 2 condition, that is nullisomy. Is it clear?
Yes, ma'am.
Okay. Now hyperploidy, that is state of having one or more greater number of chromosome than what is normal. Like if you are having 46, that will increase 47, 48, like this. So trisomy, in which you will have an extra chromosome. See, it is written here, an extra chromosome, that is trisomy. So that is denoted by 2n plus 1. Tetrasomy, condition of having two extra number of chromosome, that is tetrasomy. So 2n plus 2 condition, that is two extra number of chromosome, that's why it is written here 2n plus 2. And trisomy, 2n plus 1 condition. Now numerical abnormalities, as we have seen aneuploidy, that is variation in the number of particular chromosome within a set. So there are two types of aneuploidy, hyperploidy, hypoploidy. Hypoploidy we have seen, hyperploidy we have seen. Hypo, loss of chromosome or chromosomes. So monosomy 2n minus 1 condition. If there is a double monosomy, so you have to write there 2n minus 1 minus 1. Nullisomy, that is 2n minus 2. Hyperploidy, that is trisomy 2n plus 1 condition. Then double trisomy 2n plus 1 plus 1. Tetrasomy 2n plus 2. Now polyploidy, condition in which the cell have more than two homologous set of chromosome, that is triploid 3n, tetraploid 4n, and pentaploid 5n. Now, as you can see, this is a chromosome composition in the normal person. So it is diploid in nature. These chromosome, they are two in number, two in pair, so you can say two sets. But if there is a polyploidy, so the number or set may increase, that is 3n in condition. As you can see, 1 2 3, they are 3 in number, 3 in set, so that is triploid. If they are 4 in number or 4 in set, that is tetraploid. So now it is variation in euploidy condition. If there is a variation, it is polyploidy condition. Now aneuploidy, that is, you will see that trisomy, if there is a trisomy in second chromosome, so second number of chromosome, so that trisomy means 2n plus 1 condition. See, this is the first chromosome, this is the second chromosome pair, so you will see one extra chromosome is there because it is trisomy, so 2n plus 1 condition is there. If we are saying monosomy in first chromosome, so here you will see 2n minus 1 condition, monosomy in first, so 2n minus 1 condition. This point is clear? Hmm?
Yes, ma'am.
Okay. Now we are going to see some syndrome that is related to these. Okay. Now we are going to see some syndrome that are very important, that are related to this chromosomal aberration, and they are asked in various examination. So firstly, we are going to see the Klinefelter syndrome. That is because of having aneuploidy of sex chromosome. So you will see 47 in number and XXY condition. See, it is, as you can see, these are the sex chromosome. So here there is X, X and Y chromosome. So that this Klinefelter syndrome that occur in 1 in 500 to 1000. They are sterile, no obvious facial dysmorphy, narrower shoulders and wider hips. Then Klinefelter syndrome, they are caused by non-disjunction of sex chromosome pair during meiosis. They have an extra chromosome, extra X chromosome, so 47. And these patients show trisomy in sex chromosome. Now, affected individuals, they are actually males. What may be the symptoms? Several feminine characters like sparse body hair and enlarged breast. Their testicles remain small. Their voice may not as deep as their voice may not be as deep and have an abnormal body proportion. So for treatment, they can be given testosterone for sexual development